There we go. All right, welcome everybody. Let us get, come on. There we go. All right, so today we're going to be continuing to talk about chapter 2, okay? But today we're going to be talking more about macromolecules, okay? But we will talk about pH because I forgot to go into that last time. And then also we'll be talking about heat, okay? But I'm going to work those into the discussion of macromolecules, specifically proteins. Okay. All right, so this is what we're going to cover today. We're going to go over this week's learning objectives. I'm going to open it up for questions. We're going to go over the 4 main classes of biomolecules, the functions of those biomolecules, and the structures of the biomolecules, and what to do for next time. All right, so this week will provide a good foundation to be able to meet the following learning objectives.
Oh, it started. It's something that, like, I listen, but I also do other stuff.
Okay, so identify microbial structures and connect the structures to their functions. Identify necessary components and processes in microbial metabolic pathways. And identify key agents and processes microbial information transfer. And obviously, I did not delete that duplication there. All right, so questions. Any questions today before we get started?
I do have a question, actually, real quick.
Yes.
When it comes to hydrophilic molecules in a water solution, are they creating hydrogen bonds or polar covalent bonds, or both when they dissolve?
Good question. Let's go over that. That is a very good question. Let me go ahead and clear my drawings here. Okay, so very good question. So let's go ahead and let's get some water going here. And let's go ahead and we will put a sugar. We'll do glucose because it's fairly quick. And I don't remember if the OH goes up or down. Okay. So these bonds here and here. Okay. Well, actually not there. Let me get rid of that. That is incorrect. And the bond I didn't show here. Okay. These are all polar covalent bonds. Okay. And they hold the molecule together. They hold the hydrogen to the oxygen. Okay. So let's go ahead and put these bonds here just really little. Okay. So those are polar covalent bonds. The oxygen and the hydrogen are sharing electrons. Okay. So if we blow up the oxygen really big, remember in its outer valence electron shell, it has 6 electrons, okay, and it wants 2 more, okay, and I'm going to draw just one hydrogen, just to make it a little easier. So hydrogen has 1 electron in its outer valence electron shell, and it wants 2, okay. So for a covalent bond, okay, these electrons are being shared, okay. But because oxygen is a whole lot bigger than hydrogen, okay, the electrons tend to spend longer around the oxygen than the hydrogen. They're still sharing those electrons, okay? So they're still sharing. It's just unequal. Okay. Yes, Michelle.
Sorry, I got a new computer. I can't figure
Yeah,
this
it takes a
mute
bit.
stuff out at night. Anyway, when you say that they have, but they want, what do you, I don't understand that. Are they not hydrogen unless they get that other one?
Good question. So they are hydrogen, okay? They are still hydrogen. Even if this electron were to leave the hydrogen, we had just the proton, and we show that as H plus. Okay. So when we talk about pH, we're going to talk about molecules losing their hydrogen, but that electron, instead of going with the hydrogen, hangs out with the oxygen. So the hydrogen is still a hydrogen because it has one proton. Okay. The oxygen is still an oxygen because it has 8 protons. Okay, so we're going to put 8 plus here. We're going to put 1 plus here. Now that looks like another H. Okay. But it's supposed to be 1 plus. Actually, let me write that better so it's a little less confusing. Okay. But as far, and that still looks like another H. Excuse my handwriting. There's still those atoms. Okay. But what's happening with the electrons is why they form the associations with other atoms that they do. Okay. So because hydrogen, I'm giving the atoms human emotions to make it a little easier to understand. They don't have human emotions. It's just what they do. It's more stable to have 2 electrons in this outer orbital. It's more stable to have 8 electrons in this outer orbital. And so that explains what's happening and why H2O is a very stable molecule, okay? Because those outer orbitals are being shared, okay? They're filled. And so we can't put another thing in here very well unless we take that hydrogen out, okay? When we put these OHs on a sugar, we have to take a hydrogen off, okay? Because oxygen can only form 2 bonds.
Oh, okay.
Hydrogen can only form 1 bond.
Oh, okay. Okay.
Does that help?
Yes, yes, a lot. Thank you.
Yeah. Okay, so those are the associations that are formed, okay, by the atoms in the molecule. Okay, and those are very tight, strong bonds. Okay. So another way to think of it by giving them human emotions, let's say this is a company. Okay, these guys are all the founders of this company. Okay, and they can go out and interact with other companies, and they're going to form temporary associations with those other ones, but they have contracts with each other, and it takes a lawyer to break them. But if this hydrogen is not happy, it can go over and it can break this contract because it's not held as strongly. Okay. It says my internet. There we go. My internet connection is now more stable. Whenever I see that little warning, it means that my image on your end has frozen. So because this is a polar covalent bond and there's unequal sharing, it's easier for this hydrogen to leave. Okay, but once that hydrogen leaves, then these guys are held more closely, okay, until they get put on another molecule. Okay, but it takes energy to break that. So, but let's get back to hydrogen bonds. Okay, and I'm going to draw hydrogen bonds as green. Okay, so actually, before I do that, so I've got slight positive charge here, slight positive charge here, slight negative charge here. Oops. And the same thing is going on over here. I've got a slight negative charge here, slight positive charge here, and the same thing on all of these OH groups. Okay, oops, that's supposed to be negative, slight positive. Okay, so what happens is it's like a corporation. Okay, or the analogy I used last time, the square dancers. Okay, we're forming temporary associations. Okay, so with water, this slight negative charge kind of gives static cling, okay, with another water molecule. This slightly positive hydrogen is attracted to this oxygen. So when sugar or salt is put in water, we formed these hydrogen bonds, okay, that allow the sugar to interact with the water, but they're temporary. Okay, they're like static cling. When I pull, my clothes out of the dryer, if I forget a dryer sheet, my socks are stuck all over my shirts and my pants. Okay, but I can peel them off. When I shake it, they don't come off, but I can peel them off fairly easily. My buttons are tied on. Okay, I can't pull those off without getting the scissors. Okay, so these polar covalent bonds are like the buttons. Okay, they're sewed on there. I can take them off, but it takes scissors. Okay, these are like static cling. Okay, they're fairly strong, but they can be peeled apart fairly easily. Okay, did that answer your question about the difference between the polar covalent bonds and the hydrogen?
Yes. Yep. Thank you.
Oh, you're quite welcome. Now, while I'm here, I forgot to mention heat last time. Okay. These molecules are not just sitting there. They're bumping into each other. They're moving around all the time. Okay. So if I have a glass of water, okay, and for the sake of time, I'm going to draw the water as little blue dots. Okay, I've got a bunch of water in this glass. Okay, and they're all bumping into each other. Okay, this one's going to come over here and ricochet off. That's going to cause this one to come over here and bounce onto the side of the glass and ricochet off. Okay, they are all moving and bouncing around. Okay, how fast they're moving is what we call heat. Okay, temperature is a measure of heat, which is the movement of molecules. Okay, that's my definition and I'm sticking to it. You're going to get a more accurate definition of heat later, but this is good enough to start out with. Okay, so if something is hot, the molecules are moving around faster. They're bumping into each other more. Okay, if it's cold, they're not moving a lot. Okay, they're kind of sluggish. Okay, so if we add something like, let's go with this being a sugar cube. Okay, and I've dropped that into the water. These molecules are going to start interacting with that sugar cube. Okay, the water is going to bump into it, and that's going to knock a sugar molecule off of the sugar cube. Okay, and it's going to go zipping around. Okay, and it's going to run into water and it's going to bounce back and knock some more off. And in the process of time, because of the movement of the different molecules, okay, that sugar is going to dissolve and it's going to mix with the water. Okay, because everything is moving around and bumping into each other, okay, and jostling everything around. And because they can form hydrogen bonds, they're going to mix, okay? Now, if I pour oil in here, okay, we're going to add some oil. These oil molecules can't interact with the water or the sugar, okay? So as they're bumping around, they don't form these hydrogen bonds, okay? So they kind of get squeezed out, and because oil is lighter than water, they're all going to end up at the top of the water, because in the process of bouncing around, they're not going to form any hydrogen bonds, and they're going to end up being excluded from the water, okay? So they'll start out as globs that, because they're not interacting with these guys, they kind of start beading together, and they end up coming this direction, okay? All right, did I just flip your brains inside out?
Technically,
Okay.
wouldn't the oil only do that after it cooled?
You are, well, you're right, because like if this were boiling water, the oil is not going to have a chance to
Right.
form that layer on the top. It
Okay.
has to cool down, because they're bumping around and those oil globules are being broken up, but they never really mix.
Okay. Thank you.
Yeah, does that answer your question, Hasana? Hasada, I think I said your name right. Not
Yeah,
really.
Hadassah.
Hadassah. Okay, sorry. I will practice. I will practice.
No, you're good. Thank you.
Okay, other questions? All right, if you think of some more questions, let me know. I'm happy to stop and go back. All right, so next we're going to talk about the four main classes of biomolecules. Yes, let's see. Sherry.
Well, yeah, I just had a quick question on the test. Is it going to
Oh,
look
sure.
like this, or is it going to be more in question form, like the little quizzes that you give us and that type of thing? Or are we going to have to identify pictures, visuals?
Good question. So, to answer your question about the exam questions, they're going to be multiple choice or matching. I don't do true false very often, because personally I despise them. It's really hard to write a good true false question, and it's really hard to answer true false questions. So there'll be multiple choice and matching. Okay. But
Thank you.
oh, you're quite welcome. But they're also going to be questions where they might be what I call remembering questions, where you just have to, I give a definition of a carbohydrate, and you have to pick carbohydrate out of, from carbohydrate, lipid, protein, or nucleic acids. Okay, so fairly straightforward. I'll have some that are a little bit more difficult, where I've changed up the definitions and to show that you understand the definition. And then I'll have harder application style questions, where I give you a picture of a macromolecule that you haven't seen before, and you've got to recognize it. I'm really not trying to be mean, but it's showing your level of your grasp and be able and ability to use the information. Okay. And we'll have some practice questions today that'll be in the harder style. So generally students do pretty good with straight memorization. They do pretty good with the understanding. It's the application ones that are harder. So that's why I have you practice that one in class.
So you're going to want us to memorize what each element or I can't really think right now. Uh, we'll say carbon monoxide. What that
okay.
looks like, you're gonna have that drawn up on a test, and we're gonna have to recognize what that looks like, and then pick carbon monoxide or another harder element. Or-
Sort of. I'm not going to have you memorize the periodic table. I do want you to know the more common atoms, okay, that are found in chemistry. Okay, so they are carbon. Okay, so I'm going to do carbon as C. So that's carbon. You'll need to know oxygen, hydrogen, because to have an organic molecule, and in chemistry, when we say organic, we mean carbon bonded to hydrogen. We don't mean grown without chemical fertilizers. Okay, that's a different but still correct meaning of organic. Okay, and you're going to need to know nitrogen.
So you're not gonna draw up,
So I'm not going to what?
you're not gonna draw up this big old concoction and say, oh, that's a polymer, or that's a
You
hydro.
will need to know polymers, yes. You will need to know the four main classes of biomolecules, which are all composed of mainly these four elements, okay, these four atoms, okay. So just as a preview of what we're going to talk about, so the four main biomolecules, okay, three of them are macromolecules. In other words, they're big molecules that are made of smaller subunits. They kind of click together like Legos, okay? So you will need to be able to recognize carbohydrates. And will you need to know specifically, say, glucose from sucrose? No. But I do want you to look at, a picture and say, oh yeah, that's a carbohydrate, versus, oh yeah, that's a lipid or a protein or a nucleic acid. Okay, so you're going to need to know these four, and you're going to need to know what makes them up, what atoms go into making these guys. Okay, but I'll walk you through it. I'll make it as easy for you as I can. I realize it's complicated. Okay, other questions? Yes, Lily.
Will we have a study guide for the exam? Or is
Yes.
it just like we need to read through the textbook?
And you do need to read through the textbook.
Well,
Yeah.
yes, but
Yeah. So I do have a study guide. So if you go into Blackboard, there's a link in that dark gray menu off to the left that says study guides. Yeah. So it'll guide you in your reading of each of the chapter in the textbook and let you know what I find most important and what could be on the test. Yes, Brandon.
While we're on the subject of the test, I think it was in the practice test last week. You had to for something for identifying tuberculosis. On the test, is there going to be like any of the specific examples in the textbook that we should know, or is it going to mostly just be stuff, just the general like understanding of the material?
If I use something specific, then I will, for example, with tuberculosis. At this point, if I have a question that has tuberculosis in it, you will be able to take what you learned from the chapters and apply it to tuberculosis. I will give you the information that you would be able to do that. Did that answer your question?
Thank you. Yep.
All right. Okay, so tell you what, let's go ahead and go over the 4 different major macromolecules. Okay. So I listed them originally in alphabetical order, but I'm going to go over them now based on complexity. Okay, so we are going to start with lipids. Okay. All right. So last time we didn't talk about carbon much, okay? Just because of lack of time. Okay. But for a molecule to be organic, and in this class, when we talk about organic molecules, originally we were talking when they first defined organic in chemistry back in the 1800s. We'll say the 1800s, it could have been earlier. They were saying chemicals that came from living things. Okay, so if I take a tree and chop it down and make wood out of it, that's an organic molecule. It came from something that was living. Okay, sugars that come out of plants, they would say, okay, that's organic because it came from a living thing. Okay, then we ran into problems with things like CO2. Okay, because that comes from living organisms, but we also find them coming out of volcanoes, okay, which are not living. They sometimes act like it. It seems like they're living and they're out to get us, okay? But we have since defined organic, when we're talking chemistry, as carbon being attached, whoops, I'm jumping ahead, being attached to hydrogens, okay? So remember, these lines stand for covalent bonds, okay? So hydrogen, because it has 1 electron that it can share, but it wants 2 electrons in its outer shell, its outer valence electron shell, it can form 1 bond. Carbon can form 4, okay? And sometimes it can form 2 bonds with another carbon. So I've got to get rid of one of these hydrogens because that would make 5 and it doesn't have room for 5. Okay. So all of this represents an organic molecule. Okay. Now, because carbon is significantly smaller, okay, than oxygen, that's my story, and I'm sticking to it. I'm oversimplifying. Okay, because it's significantly smaller, okay, it has equal sharing. Okay, so these covalent bonds are equal sharing. So just as a review, what kind of covalent bond is this where we have equal sharing? It's okay if you don't remember. The effort to remember is a good study technique.
Is it a non-polar covalent bond?
Very good. Non-polar. Okay. Good job. So all of these are equal sharing. So there is no charge on this puppy. Okay. So I'm going to put a little 0 here and I'm going to put a little 0 there. Okay. So all of these, okay, are non-polar covalent bonds and they have equal sharing. Okay. And that's what lipids are. Okay. Lipids have mainly or all carbon and hydrogen. Okay. So they are mainly carbon and hydrogen. Hydro, I'm leaving a G out. All right, I'm not going to stop and figure out where I left the G out. All right, so can this dissolve in water? Okay, is it hydro? I did it again. Let's try to spell it correctly here, Teresa. spelling is not my thing, in case you couldn't tell. Is it hydrophilic or hydrophobic? Is it going to dissolve in water?
It's hydrophobic.
Very good. It's hydrophobic. Okay. It is not going to dissolve in water. Okay. So examples of lipids, okay, are oils, fats, okay, steroids, and waxes. Okay. We kind of dump everything in that is a large molecule, a large organic molecule. It's got a lot of carbon and hydrogen, and it's hydrophobic. Okay. So we tend to dump a bunch of really different molecules in. So let's go ahead and look at a steroid. Okay. So for each of these points, okay, I have a carbon I'm not drawing, because if I drew all the carbons and hydrogens, you can see that starts to get pretty messy. Okay. So I'm showing you the shape of the molecule. So steroids are 4 carbon, I think. 4 carbon, and no, I think they're 5 carbon rings. Anyway, we've got one that's up here, okay, and then I've got another one up here that's smaller, okay? But you're not going to see a lot of oxygens, okay? So generally, how you see them, okay, I'm going to take my carbons out, are these shapes, okay? Or occasionally you'll see something that looks like this, okay? One lonely little oxygen. Okay, so they're mainly going to be hydrophobic. Yes, Logan.
So you're probably going to talk about this, but phospholipids, since they have the ability to be slightly polar, is that they're still considered lipids though, right? Yeah.
Yes. Yes, and that is an excellent lead in, Logan, because these are considered very straightforward lipids. Like this would be considered a simple lipid. Steroids, they're mostly carbon and hydrogen. They have a little bit of oxygen, okay? But we have complex lipids, okay? And one that I'm going to draw for you, or parts of, is a phospholipid, okay? Which, like Logan said, parts of it are hydrophobic and parts of it are hydrophilic. So the term we use for that is amphipathic, okay? When I draw that, I'm going to write amphipathic next to it, okay, just to reinforce that. So phospholipids, which are a major component of cell membranes, okay, have these fatty acid tails or legs, okay. So I'm not going to draw them all. There can be between 12 and 15 carbons with a bunch of hydrogens attached. Okay, I'm not going to draw the hydrogens for the sake of time, but do realize there's a bunch of hydrogens here. And on this leg, we've got a double bond, and that makes kind of a kink. So it's kind of like I've got my leg kicking out here. Okay, so it looks like that. And so all of this is hydrophobic, but it's a fatty acid. So on this top part, I'm going to have an OH. Okay, and I'm going to have a double bonded oxygen here. Okay, this is an acid group. Okay, where I have a carbon bonded, double bonded to an oxygen over here, and then I have an OH group. So it's like I took water and I took one of these hydrogens off and slapped it onto this carbon. Okay. We call this a hydroxyl group, but when it's together like this, it's an acid group. Okay. So with a dehydration reaction, I'm going to attach that, okay, to glycerol. Tell you what, I'm going to get rid of this one here, just so I have room to draw. Okay. And I've got a glycerol, okay, that's got a bunch of OHs on it. Okay, a bunch of hydroxyl groups. So what I'm going to do in the process of a dehydration reaction, I'm going to take this, okay, and remove it, and I'm going to take this hydrogen and remove it, and I'm going to remove the equivalent of water, okay? And what that's going to do, I'll show you the finished reaction over here. Okay, this glycerol is now attached to this fatty acid, okay? So I've got these two phospholipid legs hanging down. On this one, what I'm going to do is this can rotate too. Unless there's a double bond, they can rotate, they can move. Okay, so these legs can kind of kick around, okay, as other molecules run into them. Okay, so I'm going to rotate this puppy up. And what I'm going to do is I'm going to attach what is called a phosphate group. Okay. And it's a big, scary molecule that I'm not going to bother to draw, but through a dehydration reaction, I'm going to attach a phosphate group. Okay. And some other molecules, but we're mainly interested in the phosphate group. Okay. And I'm going to abbreviate that as a P with a circle around it, but it's negative. Okay. So this part, okay, down here does not interact with water. Okay, actually, let me draw that further down, because that part would actually interact with water. So from here down, okay, this part is hydrophobic. Okay. This part up here is hydrophilic. Okay. So this part can dissolve in water and this part can't. Okay. And so what happens is with these phospholipids, here I'm going to get rid of my steroid here. If I put them in water, what they're going to do is the heads are going to line up with the water. And their tails are going to go inside, away from the water. Okay. They're being excluded by the water. Okay. So I've got water out here, no water on the inside. Okay. And when we're talking cells, these amphipathic complex phospholipids, okay, instead of forming little globs, okay, instead they line up like this. And we get what we call a phospholipid bilayer. Okay, so you can see my cartoon little legs hanging down or poking up. The hydrophobic legs are interacting with each other. The hydrophilic heads are interacting with the water on the outside of the cell and on the inside of the cell. Okay, cool, right? Okay, questions on phospholipids? This is a preview for chapter 3 that we're going to cover next week. Yes, Brandon and then Michelle.
Yeah, so these would be compounds, right?
Yes.
Okay.
So it has
to be a compound to cook.
Well, they are molecules because we've got more than one atom hooked together, okay, and more than one kind of atom, okay. Are they a compound? Let's go with macromolecules instead, or in the case of lipids, biomolecules. Okay. Does that answer your question?
Yeah.
Yeah, let's go with that. In this class, we're not going to distinguish between elements and compounds and that kind of thing. We're going to stick with atoms, molecules, and biomolecules, which are big molecules.
This is Michelle.
Hi.
So I guess what I'm wondering is, so their tails all go inside, and what's the point of all that? Why does that happen? Is there something special? I mean, I don't, you know what I mean? I can see why that it does it, but why does it do it?
So it does it because of these tails can't interact with water. They
Right,
can't form hydrogen bonds.
right.
And so water excludes it. But because we've got a negative charge on these phospholipid heads, they can interact with the hydrogens on water. And so they're not excluded from the water. Now, oh, go ahead.
And that makes them available to make other things?
Yes. So it makes them available to make cell membranes. Okay. And we're going to talk about cell membranes next week. But the whole point of a cell membrane is to keep the outside stuff out and the inside stuff in. So think of this as kind of like a sandwich bag. I put my sandwich in here, and it doesn't matter if my water bottle in my lunch container leaks. It's not going to get in and get my sandwich wet. And that's kind of what a cell is doing, is it wants to keep its inside stuff in. It doesn't want it dissolving away into the rest of the environment.
Okay, thank you.
Yeah, does that help?
Yes.
Yeah. Okay, so how are we doing on lipids? We're doing all right? Oh, yes, Brandon.
Just forgot to lower hand.
That's all right. Yeah, it used to be in Zoom that if you waited long enough, your hand would go down. Now they've made it that you have to put your hand down. So, all right, so let's go ahead and go on to carbohydrates. Okay. So just for the sake of room, I'm going to clear these out, and I'm going to put carbohydrates up. Okay, carbohydrates, also affectionately known as carbs. Okay. So let's go ahead and take a simple lipid. Okay. 1, 2, 3, 4, 5. Okay, I've got 5 carbons. And I'm going to put a bunch of hydrogens on it, but I'm not going to draw them. Well, I'm going to draw them in. Okay, so I've got a bunch of carbons and hydrogens. I'm hydrophobic because these are all what kind of covalent bonds? All of these are polar or non-polar?
Polar.
Are that was a good answer, but it's incorrect. Okay, it's easy to get polar and non-polar confused. Okay, so they are non-polar. That's why I keep asking, is because with repetition, eventually, we'll keep them all separate. Okay. So what I'm going to do is I'm going to take off hydrogens all on one side. Okay. And what I'm going to do is I'm going to add what are called hydroxyl groups. So it's like I took water and took a hydrogens off and attached it to this carbon. Okay. Now this bond is what kind of covalent? Polar or non-polar? Unequal sharing
Polar.
between the oxygen. Sorry. I thought I heard an answer. We're all shy now. They're polar. Okay, there's unequal sharing, so I've got a little bit of a negative charge here, a little bit of a positive charge. Whereas on these guys, because there's equal sharing, there's no charge. Okay, so I'm going to put a 0 here and a 0 here. Okay, and I'm going to do that down the line. Is this molecule going to be able to dissolve in water? Is it going to be hydrophobic or hydrophilic?
Will it be hydrophilic?
It will be. Very good. Okay. And because I've got so many OHs, the fact that we don't have a charge on this side is overwhelmed by the charges on this side. So this molecule is not amphipathic. Okay. So if I count up the carbons, I've got 1, 2, 3, 4, 5 carbons. Okay. And in a molecular formula, I would write that as C5. Okay, because I've got 5 carbons. And I've got 1, 2, 3, 4. I missed an oxygen somewhere. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, I just counted wrong. So I've got 5 carbons or 5 oxygens, and I need to move this over because otherwise. Okay. Now, if I count hydrogens, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. I've got 10 hydrogens. Okay. If I reduce this down, okay. Oh no, math. If I divide everything by 5, that reduces to CH2O, which is why we call these carbohydrates. They're carbons that have a water attached. See, I've got H2O on each of the carbons, okay, except for this one. Okay, so all carbohydrates have carbon, hydrogen, and oxygen, and I have twice as much hydrogen as I have carbon and oxygen, okay? And they are hydrophilic. Okay? So I can get simple carbs like this, and if they get long enough, they can flip around, and this oxygen is going to come talk to this hydrogen, and I'm going to get a molecule, in this case, that looks like this, okay? And one of the carbons is going to stick out. Okay, so whether you see it shown like this or like this, look for a bunch of hydroxyl groups, okay? That is one of the characteristics of a carbohydrate is I'm going to have a bunch of these OH groups. Okay. Okay, how are we doing? Yeah, okay. So I can hook these together through dehydration reactions. Okay. Using an enzyme, I can take this OH group and this hydrogen and remove water, okay? And I'm going to get something that looks like this, if I were to draw it out, okay? And so I can keep hooking those together, okay? Each of these would be called a mono. Whoops. Let's try this again. A monosaccharide. Okay, so you're probably familiar with saccharide. Okay, that's another word for sugar. Mono for one. This over here would be a disaccharide. If I keep hooking them together until I get a whole long string, that is what we call starches. Starches, if it comes from plants, okay? We have something similar in animals called glycogen. Okay. So those would all fit under the category of carbohydrates. Okay. How are we doing? Yeah, if you haven't been exposed to this before, okay, it can be really confusing. Remember, you need at least on average 6 repetitions of the material before your brain is going to move it from short-term memory, easily dumped, memory to long-term memory. So in other words, it's going to take about 6 repetitions for it to stick. Okay. So try not to get frustrated. Okay. Yes, Sherry.
So could we safely assume that if we stick to the study guides that you've provided, that those will cover all the material that's going to be on the upcoming big test?
Yes. But
Thank
what
you.
I would recommend, and this is what works for me because I'm a visual kinetic learner, I would draw it out. Okay, especially for these structures. You're going to draw it out, and I would do it from memory. Okay, so look at it in the book, close the book, okay, or blank your screen, and then try to draw it out. And you're not going to get it right to start out with. You're going to forget a lot. That's just a part of learning. And so then you go back to the book and you say, oh, I forgot this, or I got this wrong, and then you fix it. And then you get a new page, you wipe off your dry erase board and try it again. Okay. For me, that's the most effective way to do it. Okay. If you're more of an auditory learner, closing your eyes and kind of talking through it might work better. Okay. But yes, the study guide is a good guide for what's important.
Thank you for those, by the way, very
Oh, you're
much.
quite welcome. Okay, tell you what, let's move on to nucleic acids. Okay. All right, so nucleic acids. All right, so I'm going to get my 5-carbon sugar here, and I'm going to draw it kind of skittywampus. Okay, now remember, carbohydrates, I've got a bunch of sugars here. Or excuse me, yeah, duh, this is a sugar. I've got a bunch of hydroxyl groups, a bunch of these water groups stuck on there, okay? So this is a sugar. Okay. Now I'm going to modify this puppy to make a nucleic acid. Okay. So the first thing is through a dehydration reaction, I'm going to add a phosphate group. Now, normally with the phosphate groups, okay, we tend to abbreviate them as this, but this is the actual structure. Okay, see all those hydroxyl groups? Okay, we also abbreviate it PO4H, either 3 or 4. I'm going to put 3 because that's what I've got up here. Okay. So is this going to be hydrophilic or hydrophobic? Hydrophilic, because it's got a bunch of these water groups. Okay. So through a dehydration reaction, I'm going to remove a hydrogen and an OH group to remove the equivalent of water. Okay, and I'm going to hook that on there. Okay, so that I get something that looks like let's change colors. Okay, like that. And let's go ahead and put my hydroxyl groups here, my water groups. Okay. Now, over here, I'm going to put another group that we call a nitrogenous base. Yes, Michelle. Let's see, you're still muted.
Sorry.
Oh, no
Um,
problem.
What's the difference if you write it OH or HO?
It's no difference. It just depends upon whether there's room on the drawing.
Oh, okay. Thank you.
Yeah. I know I flip it around, and it's because in solution with all of these atoms and molecules bumping into each other, they do rotate around. Okay. So these single covalent bonds, they can twist and turn. So sometimes they might be facing this way, sometimes it might flip over this direction. Okay. All right, so for a nitrogenous base, I've got either a 1 or 2 ring. Okay. On this one, I'm going to draw 1 ring, and it's got a bunch of nitrogens on it. Okay, and it might have a double bonded oxygen. Over here, I'm going to put a double ring. Okay, and I have a bunch of nitrogens. That's why we call it a nitrogenous base. Okay, so nucleic acids have all 3 of these. Okay, they have a sugar group, they have a phosphate, sometimes more than 1 phosphate, and they have a nitrogenous base. Okay, or sometimes we just call it a base. And like the carbohydrates, we can snap these. And by the way, we call this from here to here a nucleotide. Okay. So through a dehydration reaction, I can hook these together. Okay, I can take this off and this off. Whoops, I took my oxygen off. I don't want to take my oxygen off. And I can hook these together. And when I get a big long chain, that makes a nucleic acid. Okay, so nucleic acids are composed of carbon because all organic molecules have carbon. They all have hydrogen. They have oxygen, and they have phosphate, and they have nitrogen. Okay, so when you're looking to identify a nucleic acid, you want to look for the sugar group the nitrogenous base in this phosphate group, okay? And we're running out of time. Let me do proteins real quick. Okay, so proteins look a little different than those others. I started with lipids, okay, which were just carbon and hydrogen. I added some OH groups onto there to make carbohydrates, okay. I took a carbohydrate, added a phosphate group and a nitrogenous base, made a nucleic acid, okay. Next, I'm going to take a slightly different shape, okay, different monomers, okay, for proteins. Okay, so I'm going to take a nitrogen and a carbon, 2 carbons actually, and I'm going to put an acid group on there, okay? So this double bonded oxygen with a hydroxyl group is an acid, okay? Like on the fatty acids. And then this group on the end is what we call an amine, okay? So I have an amine group and an acid group, and that makes an amino acid, okay? Which is what we use to hook together to make proteins, okay? Now, this first carbon always has a hydrogen sticking up, but here I can put all sorts of groups, okay? It just depends upon which amino acid it is. So we call this the R group, okay, or the side chain. And I can put all sorts of different kinds of side chains. I can put an H here, okay, which would make this side chain hydrophobic, okay. I can put an OH, which would make it hydrophilic, okay. So these R groups or side chains can be either hydrophilic or hydrophobic, okay, phobic. That doesn't look right. I left an H out. Okay, and it all depends upon how much oxygen is there. Okay, so remember philic, you have oxygen or nitrogen. Okay, hydrophobic, you've got a lot of H's and C's. Okay, so if I had a side chain that looked like this, okay, remember each of those points is a carbon, that would mean it was hydrophobic. Okay. If I add an OH group, it's mostly hydrophobic with a little bit of hydrophilic thrown on there. Okay. All right, so I'm going to start hooking these together, these amino acids, to make a protein. Okay. So I'm not going to put the R group in. We're just going to leave it as a question. Okay. So now I'm going to do a dehydration reaction. Yes, in biology, we do a lot of dehydration reactions to put things together. So I'm going to take this OH group off of this end, and I'm going to take this hydrogen. I'm going to remove the equivalent of water to hook these guys together. Okay. And so I'm going to keep doing that to make a protein. Okay. Oops, let's get rid of that. Okay, and I can keep hooking these on, okay? Because proteins can get pretty big, okay? So the amino acid itself, this part of it is hydrophilic. This part can be hydrophilic or hydrophobic. So depending upon the amino acid, that makes amino acids what? Okay. Oh, we don't have time to go into protein folding. Tell you what, next time we meet, I will save some time to go over that. Okay. But I want you to read about it in the, what do you call it? In the textbook, okay? Protein folding, okay? Because of protein folding, this chain, this line can be folded into all sorts of shapes, okay? Which means that proteins can form about any shape, okay? So we find a lot of proteins in cells because they can form a whole bunch of shapes, which means they can perform a whole bunch of functions, okay? And I especially want you to read about denaturing, okay? Yes, Candace.
What are your office hours this week? Because I think I read, did they change or?
Yes, so on Monday, I switched that office hour to Friday, so it's gonna be from 1:30 to 2:30. Today, I'm gonna have office hours from 1 to 2. Normally, they'd be from 1:30 to 2:30, but I have a meeting at 2:30.
So 1 to 2 today.
Yeah,
Okay.
1 to 2 today.
Thanks.
Come on. There we go. All right, let's do a quiz real quick. And we're going to skip past these, okay? Let's skip past the functions. I'm gonna have you read that one because that's a less difficult one. Okay, so for this first one, I'm gonna pull up a poll. And I was gonna set up a 4 multiple choice option, but I did not. Okay, so ignore E, okay? Is this a carbohydrate, A, a lipid, B, a nucleic acid, C, or a protein, D? Ooh, we got some good voting. And it's okay if you don't know, that's why we're practicing. This is a good study technique. Remember, you got to go over this, if it's brand new information, at least 6 times before it sticks. Okay, we've got about, oh, here we just broke 60, 64. Okay, 72. Okay, just for the sake of time, because I want to do at least one more quiz, I'm going to end the poll and share it. Okay, so we have a tie between carbohydrate and lipid, which, good job, especially if this is the first time you've heard this, okay? Carbohydrate and lipid are your best options. So if you can eliminate nucleic acid and proteins, that is half the battle on a multiple choice question. You can generally eliminate it down to 2, okay? Now we got to decide which one is better, carbohydrate or lipid, okay? So, are there a lot of oxygens? Do you see any oxygens on there? There's one. There's one lonely little oxygen. So, which one's better, carbohydrate or lipid? I'm going to relaunch the poll. And I want you to vote again between carbohydrate or lipid. Oh, and we've still got even voting, which actually at this point is not bad. Okay, because remember, you've got to have more than 1 repetition to remember it. Okay, I'm going to end the poll and I'm going to share the results. Okay, we got a little bit more votes, okay, for lipid, and lipid is the correct answer, okay? If it were a carbohydrate, we'd see all sorts of OH groups hanging off the edge here, okay? All right, let's do another one real quick. So I'm going to relaunch the poll, okay? All right, so here is, come on, let's get rid of this. Here's the other one, okay? So, is it an A, carbohydrate, B, lipid, C, nucleic acid, D, protein, and there's no E. Okay, for the sake of time. Oh, we've still got people voting. Okay, now I'm gonna use the poll. Okay, most people picked carbohydrates. Good job. Very good job. See all these OHs? Okay, see all these oxygens? That makes it a carbohydrate, especially if I were to go through and count and go 1, 2, 3, 4, 5, 6, and I've got 1, 2, 3, 4, 5, 6, okay, equal numbers of oxygen and carbon. It's a carbohydrate. Okay. All right. So anyway, we're gonna, I went too far. Okay, so this is what I want you to do for next time. Start working on the library badge. Okay, it is due on Friday. Finish going through the week 3 lecture folder if you haven't finished doing that yet. Fill out the chapter 2 section on the exam 1 study guide. And start case study number 1 in biochemistry, in the biochemistry batch. Okay, the biological chemistry batch. The case study 1 and the biochemistry badge are not due this Friday, but I'm giving you 2 weeks to work on the case study number 1. It's not due this Friday, but it's due a week from Friday. And the biochemistry badge, I'm giving you 3 weeks because it'll probably take you about that long. And it will help you reinforce what you're learning in this chapter. And then when it's due, it's when we're going to be talking about genetics. And it's going to be helping reinforce that too. Yes, Sharon.
So I noticed you put a note about, using the library resources and such at CWI on the case study 1. If they aren't all references from there, or if there are only a few from there, are we going to be marked down for that?
No, with case study number 1, you can find most of the information in the textbook. I do want you to reference the textbook though.
Okay, gotcha.
Yeah, if you can't find it in the textbook, then you can look online, but make sure you use what you learned in the shift badge to pick out reliable sources. Yes,
Okay.
Candace. Okay, just for the sake of time, email your question to me, Candace, or definitely come to my office hours today. Okay. By the way, everybody is free to come to my office hours. If I didn't have a class immediately after this, then I would go ahead and hang out and answer your questions.